Right. Thanks, Steve. And it's just turned 10:30. We have 63 people online. That's great. So thanks to all of you for coming along today. And I'm especially pleased to welcome our speaker, Emily Mycorrhizal. I hope I've got that pronunciation right. I've been practicing. Emily's currently at the University of Sheffield, and she's researching for a PhD in soil ecology. And her particular focus is on the fungal networks, which, although largely unseen, are such a vital part of the subterranean biomass. And she's going to tell us a great deal more about them than David Attenborough did in his Wild Isles program a few weeks ago. So without further ado, I'll hand over to Emily to lift the lid on this hidden world.
Perfect. So yeah, thank you, Barry and Steve, for organising this, and thank you everybody for being here. So I'm going to try and share my screen. Hopefully it should work. It worked on Monday when we were trying it. So let's see.
Perfect. Perfect.
Right. So let me do this. If I try and do present. Can you see the full screen now? Can you see the next slide also?
Perfect. We can see the title slide, mycorrhizal fungi.
Just the title slide.
Just the title slide.
That's helpful. Okay, then. So I'll make a start. So yeah, the title of the talk to get today, and I guess our meeting, is mycorrhizal fungi, the hidden network beneath our feet. So as Barry was saying, yeah, my name is Emily, and I'm doing my PhD at the University of Sheffield. So I'm in the group of Katie Field. So yeah, everybody in our group broadly works on mycorrhizal fungi, but as I'm going to explain, there are actually different types. So some of us work on this type, which is known as a vascular mycorrhizal fungi, and some other people are working in a sort of more recently discovered group of fungus, which is called Emphrae for short. So yeah, before I start and sort of move into the bulk of my talk, I just wanted to take you a little bit through my journey and give a little bit of context as to how I ended up doing what I'm doing. So to give a hint to my surname as well, I actually grew up in Greece. So my dad is Greek and my mum is English, and I grew up in Kalamata, which some of you might have heard because of the olives. So it's in mainland Greece. Not so much of it's becoming more and more touristy, but yeah, I think locals are quite lucky in the sense that it's quite sort of an authentic and quiet place. So yeah, I would recommend you visit if you get the chance. But yeah, once I left school, I decided to take a gap year. So I went to this place here. So this is Coutances in Normandy in France, and I was working there at an environmental education charity. So this sort of experiences there sort of confirmed my love for the natural world, let's say, and sort of decided to study biological sciences. So sort of wasn't sure whether I just wanted to be in the lab or whether I also wanted to be out in the field. So I thought, okay, I'll study quite general, which gives me options for both. So this is what I did at Aberdeen. So I went up to Scotland and I went to the University of Aberdeen, where I studied biological sciences. So overall, I stayed there for 5 years, but I did take a gap year. I took a gap year and I did what was Erasmus at the time at the University of the Basque Country in Bilbao. And then one bit, so then I went back to Aberdeen after this year, and then I graduated in 2019. An important bit that is missing from this slide is actually when I first started working with mycorrhizal fungi, which was after I graduated. I went to Berlin for a 3 months internship. So I went to Berlin because I wanted to actually be in Berlin because a good friend of mine lives there, and just thought it would make my life easier. But somehow I ended up working in one of the best known soil ecology labs, one of the best known soil ecology labs in the world. And I didn't know much about mycorrhizal fungi at the time, but they work a lot on mycorrhizal fungi, and that was basically my introduction. and I was working on sort of how microplastics and mycorrhizal fungi sort of interact with plants. So that was very good, and I was like, finally, there's, I could make up my mind, because I did biological sciences, quite a broad degree, and I was like, okay, finally, I can maybe make the decision to work more with plants and soil. So when I was in Berlin, a job came up at the University of Leeds. So that was working as a research technician. So I applied for that, and I was lucky enough to get that, and that was what brought me back to the UK. But in England, that this time was the first time I was living in England. I went to Leeds, and this is when I started first working with Katie Field. So Katie was at Leeds at the time, and then I stayed there. Yeah, soon as I started, COVID hit, and then Katie announced that she's actually moving back to Sheffield. But I stayed in Leeds, and I finished didn't finish. I actually finished early because I decided I wanted to continue sort of working on a similar project, which I'm going to explain the details of later on a similar project, but actually doing my PhD on it rather than working as a research technician. So yeah, that's when I moved to the University of Sheffield, where I'm currently at, and I'm in my second year. So yeah, I'm lucky enough. I've got 4 years funding, so I'm sort of almost halfway through. So the plan for today is shown on this slide. So I'm sort of going to go over some basics about what mycorrhizal fungi are. Then I'm going to introduce this concept, which is sort of biological markets, and I'm going to explain a little bit more about what this is and how it's used in mycorrhizal ecology. Then I'm going to go on and link this to what I actually do, or at least show you parts of it. And then finally, we're going to go on sort of talk a little bit about this term that you might or might have not heard, which is the wood wide web. Okay. So what are mycorrhizal fungi? So yeah, before I say too much about it, I just wanted to sort of ask about as to whether people have heard of mycorrhizal fungi before. So feel free to put a yes, type in a yes or a no in your chat box, I guess. One thing is that unfortunately I do not think I can see the chat box, and if I minimize, I can see it. Yeah, there it is. Don't know if can you see my chat box now?
Not sure I can at the moment. I've got it off, but
okay, I can see it, so that's fine. You can only see the slides still, yeah.
Yeah.
Okay.
We'll check that. We'll look at that at the end, Emily.
Yeah, I can see the answers coming in. So yeah, most people have heard them. Okay, using them for gardening purposes. Okay, perfect. And the next one is next question, which again you can type in yes, no, or don't know, is do you think mycorrhizal fungi are good for plants? Yeah, big yes, no, yes. Probably. It's good. I said yes, no, or don't know, but we haven't seen don't know. Okay, for most plants, yes. Yes, most scientists that work on the field would say it depends. That's always a good go-to answer. Perfect. Okay. So I'll move on. Basically, just explain. So I'm gonna go on and explain that there are actually different types of mycorrhizal fungi, but I'm gonna give the basics of what they are and what they do for anybody that might not know, using the example of arbuscular mycorrhizal fungi. So this is a specific type, and it's actually the type that I'm working with. So I'm more familiar with these guys. So arbuscular mycorrhizal fungi, they're obligate biotrophs. So what that means is that they cannot live on their own, or they cannot complete their life cycle on their own. So they associate with plants. So around 80 to 90% of land plants actually associate with arbuscular mycorrhizal fungi, and they are root endosymbionts. So they actually are shown in this sort of cute schematic here is what they do is that they penetrate the root cells and they live there, at least for part of their life cycle. And the other schematic here is basically showing one of the fundamental aspects of their symbiosis, which is that they help plants acquire nutrients from the soil. So what they do is, like, if this is the fungus and this is the plant, the fungus is providing the plant with extra sort of mineral nutrients or water from the soil in exchange for carbon in the form of sugar or lipids. So this carbon that the plant provides is carbon that has been photosynthetically fixed. There are other benefits from a plant perspective. There can be other benefits from arbuscular mycorrhizal colonization, but I think this nutritional symbiosis is the most studied one. So yeah, this is also shown here, but basically you've got your plants in the soil, and then basically because of the nature of fungi, they form very thread-like structures, which are known as hyphae. So these blue structures here represent the hyphae. So they extend in the soil and they grow, and they're finer the roots, so they can access pores in the soil that would otherwise be inaccessible to plants. And that way they can either extend the root area, or even within the root area, they can provide minerals that would otherwise be inaccessible to plants. Okay, so this slide is basically just to reiterate what I was saying in terms of there being different types of mycorrhizal fungi. So most trees actually, at least in temperate regions, they form this type of mycorrhizal association, which is called ectomycorrhizes. So these are also the ones that form mushrooms. So mushrooms are the fruiting bodies of the organisms which live largely below ground. So ectomycorrhizas, again, there's an exchange of nutrients from the soil, but via the fungus to the plant, and then carbon going from the trees to the fungus. But the only difference is that rather than the hyphae penetrating the root, they form a structure that sort of surrounds the roots. And then you've got the other general group, which is the endomycorrhizes. The arbuscular mycorrhizal fungi, which I was explaining before, fall within this category. And some trees form arbuscular mycorrhizal symbiosis, but they're mostly known for a lot of herbaceous plants form them. And then you've also got ericoid mycorrhizes, so heather and form this type of associations, and orchids also form mycorrhizal associations. And then figure B here is basically what you're seeing. You're seeing the fundamental aspect of the nutritional symbiosis, where a plant, as I was saying, can get water, phosphorus, nitrogen, or other nutrients from the soil in exchange for giving carbon to the fungus. Figure C here is what is showing that sometimes you can have one fungal species that forms two types of mycorrhizal associations. So here you've got this, I think, is a Ruzula species that forms both ectomycorrhizal associations with trees, but also orchid mycorrhizal associations with some orchid species. And you can have it the other way around. So here you've got like a tree, like poplar trees, for example, they form two types of mycorrhizal associations. They form ectomycorrhizal associations and arbuscular mycorrhizal associations. And this last figure here sort of hinting towards this idea, which will form the basis of our talk today, which is that sometimes, because of the nature of these fungi, they live largely below ground and they form these hyphae, and they have the capacity to actually colonize two neighboring plants. So here you're seeing a below ground network that's colonizing two neighboring plants. They don't have to be the same species. Sometimes they can be the same species. And this forms the basis of the debate. So when you have a fungus colonizing two species, scientists tend to call this common mycorrhizal network. And this is what has been known and popularized as the wood wide web when it's referring to forests. Okay, so moving on to explain a little bit about the idea of biological markets. So this is something that I'm quite interested in, because actually, even though research in mycorrhizal fungi is actually picking up these days, and it's quite popular and a lot of people are interested in them, actually we still are not entirely sure about the fundamental aspects of the symbiosis. So we know that plants associate with mycorrhizal fungi. We know that there's this exchange of nutrients, which is called the reciprocal resource exchange, that is shown here. But actually, what governs the amounts of nutrients being exchanged, we're still quite unsure. And obviously, different species of plants and different species of fungi respond in different ways. And then in nature, things get complicated. You have bacteria there, you have other organisms there, and things happen in terms of environmental conditions. Everything is fluctuating, and it's just, as an ecologist, it's very hard to monitor this and to sort of figure out what's happening. One thing that has also intrigued evolutionary biologists is actually how this mostly beneficial symbiosis has occurred, and how it has been evolutionary been stable. Because it's quite likely that you could have sort of cheater plants or cheater fungi actually getting carbon from the plant, but maybe not providing as many nutrients in return, or vice versa. So one idea people have proposed, this idea of the biological market, where basically what they've said is that we should view carbon and phosphorus or nitrogen in the soil as currencies to be exchanged, and sort of view the symbiosis as such. And they've done quite a few experiments where they've shown that actually the fungus and the plant can in some ways be in control of this exchange of nutrients. And when you also have two plants connected to each other, you can sort of test this. So this is shown in this slide here. So basically what you have here, you've got two plants. One, you can see here, this plant has aphids on it. So from a fungus perspective, this is not a very beneficial host, because this plant is induced to herbivore stress, and it's arguably a lot of the carbon that it's getting through photosynthesis is actually consumed by the aphids. So what you're seeing below ground is that these thread-like structures are showing the hyphae that are colonizing the two plants. The neighboring plant here does not have aphids, so you could say from a fungus perspective, this is a high benefit host. So one idea behind the common mycorrhizal network is, can you also have sort of how much agency does the fungus have, and how much control does it have as to where it allocates its resources? And can it detect that this plant has pest on it and actually funnel more of its phosphorus and nitrogen that it's harvesting from the soil and provide it to this healthy plant, because it's actually getting more carbon in return? So this is one idea that we are trying to test, and I'm doing something similar in my experiments. I'm gonna touch more on this later. But yeah, it also gets more complicated, because usually when we do experiments, we like to to some extent control for things, and to sort of you start doing things sort of in a more simple, straightforward way by using one fungal species. But here you can see like the yellow and the red threads are representing different fungal species that might actually have a different behavior. So yeah, moving on to what I do. So broadly, I am interested in soil biodiversity and how that interferes with plant and plant growth, and in some aspects to agricultural applications as well. Yep, there it is. So basically what I do is that I grow plants. So the project that I was working on in Leeds when I started working as a research technician, so yeah, this is me in Leeds, is basically we wanted to see how plants interact with different organisms in nature. So I was saying mycorrhizal fungi, not always, but in some cases they can be beneficial to plant growth. But also in nature, you don't just have plants associating with AMF. You also have other organisms. So in our case, we were interested in nematodes. So potato cyst nematodes in particular. So you can see the organisms here. So what I basically do, I grow plants, I look them under the microscope. So here you see this photo here showing a potato root colonized with arbuscular mycorrhizal fungi. And this one is showing a juvenile potato cyst nematode invading the root. So from a plant perspective, we could say that this is the friend and this is the foe. So we were just interested in seeing like who wins the competition for plant resources. Is it the friend or is it the foe? And that takes a lot of growing a lot of plants and doing a lot of cleaning of roots, and then looking things under the microscope, and then obviously as all scientists going to conferences and presenting our research. So yeah, so as I was saying, my project specifically, I'm working with potato plants, and one big problem from an agriculture perspective with potato plants in this country as well is these guys here, the potato cyst nematodes. So here you can see the cysts actually, which are the end of the life cycle for the females. And these are created on the roots, and they contain hundreds of eggs. And when the crop is harvested, the cysts remain in the soil for tens of years. And when the next crop comes in and the plant is growing, they sense the signals from the plants, they hatch and they infect the potato plant. And this is the juvenile. So once they hatch, they can migrate within the soil, they can pierce the root, and the juveniles feed and suck out all the plant resources. Here you can see some of the damage. So you can see sort of the nematodes, potato cyst nematodes in the field that can be quite patchy. So here you can have a very high density, and your yields are heavily reduced, and maybe next to that you don't have that many, and you can see how sort of the yields, even just visually, you can see these plants are not looking very happy compared to sort of a healthy bushier potato plant. So yeah, on the other hand, a lot of people have been proposing the use of arbuscular mycorrhizal fungi to maybe use it almost as a biocontrol. So you apply mycorrhizal fungi, or you do some specific management of your soil that increases the native arbuscular mycorrhizal fungi, and then you can use them to counter some of the negative effects of nematodes, for example. So we've done quite a lot of research on this, and we have basically found that yes, arbuscular mycorrhizal fungi can help with potato cyst nematode damage, However, we're not quite sure as to how long-term this effect can be, because another thing that we found is that because when you have a arbuscular mycorrhizal fungi there, the plants are healthier. Actually, they can sustain a larger proportion of nematodes. So we were seeing that although the plants were healthier, the nematodes were also doing better. So the plants could tolerate the nematodes, but after many years of unpropping seasons, you could say that you would be building up the nematode population, and eventually this effect would crash. Anyway, so for my PhD, basically what I wanted to do though was to investigate the capacity of arbuscular mycorrhizal fungi to help with nematode damage, but in a little bit more realistic scenario when you've got two plants. So these are sort of the one of the systems that I'm growing looks something like this. So I've got sort of larger container pots than we usually use, and there's a mesh down the middle. So this mesh excludes roots. So I'm growing two plants on either side, excludes roots but allows the hyphae to cross. So we sort of say that, you know, both plants are colonized below ground by the same fungal network. But then what I'm doing is one plant has nematodes. So this is shown a little bit better in the schematic here. So these white blobs are the cysts. So we can say if this plant has nematodes, and then it's neighboring a plant that doesn't have nematodes, I'm going back and sort of looking at the same ideas what I showed before with the aphids. So I'm interested in knowing, can the AMF discriminate between these two hosts and actually move more of the resources to the healthy plant? And if so, do they actually get more carbon from that healthy plant? So the way we do this is we use radioactive labels. So for phosphorus, we use 33P. I'm not going to go too much into the technical details, but basically in these pots here, we apply a liquid amount of 33P, and then we can track that within our system. So 33, the phosphorus, would be picked up by the fungus, and from the fungus, we can see how much of it ends up in the plant. And here you see some bags. So these are the plants before a label with radioactive carbon. So we bag the plants and we release radioactive carbon and allow it to photosynthesis basically to occur for, in my case, usually around 24 hours. And then again, I can see how much of this carbon has gone through the flood and down below ground to the nematodes and to the AMF. Okay, so last section of the talk is the wood wide web. So as you have seen, like sort of a disclaimer, I don't actually work with trees, and even though there are quite a few similarities, actually ectomycorrhizal fungi, you know, when you're a scientist and when you're going into the details of it, do form symbiosis in a slightly different way. But I do work with this idea of the two neighboring plants. So we've been drawn a little bit and actually are interested in this debate about the wood wide web. So similar to before, just wanted to see whether you've heard of the term wood wide web before. Yeah. No, yes, no. Okay, that's interesting. I think one thing that I find when I try and explain people what I do, yeah, one of the first things that I get asked is like, oh, trees talking to each other, and we can talk a little bit about this more if you like at the end after I've sort of presented the debate, because it's quite interesting from our perspective, because people get enthusiastic and passionate about our research. But yeah, it does come with some problems sometimes. So the next And last question is, do you believe that the wood wide web is a real thing in nature? Yes, some don't know. Yes, very nice. So let's see what others what others researching the field think. So yeah, going back to this slide, the idea. So this is a schematic, but the idea is that, especially in a temperate forest where you get a lot of mycorrhizal fungi, you have trees forming the forest, and below ground, same species or different species of fungi growing and colonizing plants. And the idea is that the same individual, which I'm not going to go into this debate, but what is an individual when it comes down to fungi can be a bit of a debate in its own terms. But basically, yeah, you can have sort of trees being linked up by mycorrhizal fungi. So there's the research on this. It's actually a lot of research from this actually started from Sheffield. Sir David Reed did a lot of work on using radioactive isotopes and trying to trace carbon from one tree to the other, or sort of a controlled system. So it's been quite a few years that research on this has been taking place. This Nature front cover is from a Nature 1997 journal. That was when the term, I think, was popularized quite a lot. It was the wood wide web term was coined. It was after Simard and colleagues presented some of their results in forest. And then from that, debate did continue, but I think just the term captivated scientists and the general public. And then there are many videos and sort of public and science communication information out there, but just thought I'd show you this one from the BBC. Some of you might have seen it before, but it's something that I've used before with students. So I'm just gonna exit with the caption.
Yes, as we can hear it, thank you.
Trees may look like solitary individuals, but the ground beneath our feet tells a different story. Trees are secretly talking, trading, and waging war on one another. They do this using a network of fungi that grow around and inside their roots. The fungi provide the trees with nutrients, and in return they receive sugars. But scientists have found this connection runs far deeper than first thought. By plugging into the fungal network, trees can share resources with each other. The system has been nicknamed the Wood Wide Web. It's thought that older trees, fondly known as mother trees, use this fungal network to supply shaded seedlings with sugars, giving them a better chance of survival. Those trees that are sick or dying may dump their resources into the network, which might then be used by healthier neighbours. Plants also use fungi to send messages to one another. If they're attacked, they can release chemical signals through their roots, which can warn their neighbours to raise their defences. But like our internet, the Wood Wide Web has its dark side too. Some orchids hack the system to steal resources from nearby trees. And other species, like the black walnut, spread toxic chemicals through the network to sabotage their rivals. Arboreal cybercrime aside, scientists are still debating why plants seem to behave in such an altruistic way. The hidden network creates a thriving community between individuals. When you're next in woodland, you might like to think of trees as part of a big superorganism, chatting and swapping information and food under your feet.
Perfect. So yeah, I chose to show you this video. I think quite fascinating. You know, it tells a good story. However, it has been picked up recently by some scientists, which have resurfaced this debate as to what is really a common mycorrhizal network, and actually how much do we really know about the functionality of it. So this, the remainder of the talk is going to be based on this paper here. So this was actually also published in Nature, although Nature Ecology and Evolution, which is a journal which is part of the Nature Singer group these days. So it was Justine Karst, Melanie Jones, and Jason Hoeksema, some prominent scientists in the field, some of which actually, I think Justine was actually working with Suzanne Simard. So, you know, they've been trying to balance their resurfacing the debate and sort of going back into the studies and looking as to how well they had been done and how much we can say about their findings. But actually, you know, they've been trying to balance that with not sort of being seen as pointing the hand to others and sort of saying like, we are the good scientists and, you know, you are not good and you're not good doing good science. It's more like, you know, everybody coming together and sort of, you know, going back to how science is done and just checking over, because they feel that this Wood Wide Web has captivated people's minds, but actually there is not enough evidence about it. So the title of their perspective article is Positive Citation Bias and Overinterpreted Results Led to Misinformation on Common Mycorrhizal Networks in Forests. And basically what they are saying is that we know that common mycorrhizal networks are a thing. You know, these fungi are real, they exist in nature, they colonize plants. What we are actually unsure of is their functionality, and sort of in ecological terms, what they are really doing. So they reviewed the literature and they found 3 popular claims about the common mycorrhizal networks. The first one, and that also included public science literature. The first one is that, so CMN stands for Common Mycorrhizal Network, which sort of, let's say it's equivalent when we're speaking of forests to the Wood Wide Web. And the 3 claims that they found is that these networks are widespread in forests. So it seemed like people seem to believe that this is a thing. The second claim was that resources are transferred through these networks. And this resource transferred led to increased seeding performance. So what you saw in this video is basically the idea of, a big mother tree and you've got the seedlings nearby and they're getting resources. And linked to this is that mature trees communicate preferentially with offsprings through common mycorrhizal networks. So these were the 3 claims that they found. And then lastly, they also found that citations, this is in scientific articles, were biased towards positive effects of common mycorrhizal networks. However, basically they went back and reviewed the actual literature, scientific literature around this topic, and they found that none of these are true. So in terms of for the first one, that they are widespread in forests, this claim has you know been said time and time again. However, too few forests have been mapped, and actually only 2 studies demonstrate actual continuity of fungal links among trees. So this is something very difficult to do, because obviously you're working with soil, you're working underground. It's very difficult technically to check, to get the hyphae from the soil and to check without destroying the network, and to see whether anything is functional and whether 2 trees are actually connected. And then it can get very technical, but again, I'm not going to go too much into the technical details. I'd say their article is quite accessible, so if anybody does feel into delving more into the technical aspects of it, do go and read the article, and they give some more examples of what that actually means. So yeah, the other thing with this is that actually too few tree species have been studied. So out of like, I think the current estimate is like there are around, I think 70,000 tree species on Earth, and most studies have been done with 2 species. So again, I think it's fair to say that, you know, you can't really extrapolate from 1 or 2 tree species and sort of just say that that's a thing out there and it's happening. You need to be careful as to how you sort of interpret your results there, basically. The next one is that resources are transferred through common mycorrhizal networks, and that lead to increased seeding performance. So the interesting thing with the main caveat with any resource experiment is that there are alternative explanations. So basically, there are 3 main ways that resources can be exchanged or moved in nature. One is via the mycorrhizal networks. The other one is through the soil, so there can be passive diffusion. And the last one is by direct root-to-root contact from one tree to the other. So this is where a lot of our experiments fall short in terms of controlling for these alternative explanations. So basically what they concluded is that the function of the common mycorrhizal networks remains actually unresolved because experiments have not conclusively demonstrated this common mycorrhizal network mediated interplant resource transfer, and more so even if there in cases where there's any transfer. So for example, in that very popular Nature paper that Simard published in 1997, the amount of carbon that is being transferred from one plant to the other is so small that arguably it sort of raises the question of what does it even mean in terms of ecological function and plant performance. So I'm not going to go into the details of this, but basically this is what they've done here. They've taken every single study that fitted the criteria of broadly working on this topic, and ways that people have tried to control for alternative explanations of carbon being moved below ground. And there's almost every study there are alternative explanations to be considered. So more experiments are needed, basically. So yeah, you can go back and sort of read, but basically I think this picture is showing you, it's giving you an idea about how people control for this transfer. So one way that the people do it, so this first one is showing, so you've got 2, so you're interested in seeing whether this plant and this plant, actually whether these 2 plants, whether there's any carbon moving from one plant to the other. And what you do is you also have another plant in the system. So if these 2 form a specific type of association, so ectomycorrhizal association, you have another one that doesn't form that type. And if any carbon ends up in here, you know it's not via the network, because this plant and this plant do not form the same network, because this one is ectomycorrhizal and this is arbuscular mycorrhizal. So you can use this as sort of control for alternative paths. The other main way that people do this kind of research is they use meshes. So they use big bags of pores with pore sizes that exclude or allow roots to attach, but that comes with all sorts of caveats about the volume of soil that you have and stuff. So it's basically just to prove that it's very hard to do this kind of research, and people that actually do it should know best. The interesting thing is, you know, scientists often come and say, oh, it's because of the media, you know, they don't pay enough attention into what scientists are saying, and they're just extrapolate from that and make it into a nice story. But I think I'll skip. Oh, will not include. I did not include the last part, which is basically going back to this bit, the citations. So what they found is that scientists themselves have taken prominent articles that have worked on this topic, and they have brushed through all the caveats or the alternative explanations that the scientists were proposing, and they have just claimed effects of the Wood Wide Web, whereas maybe it wasn't the case. So it's actually scientists themselves are also responsible for this spread of misinformation. So with that, I think I'd want to conclude by saying thank you, and sort of by saying that I think where we're currently at is that this debate, which is not new, has sort of again resurfaced. I hope you're still passionate about the mycorrhizal fungi and plant and soil ecology, and I think my take on it is that, you know, you don't need to have trees loving each other and sharing resources altruistically to actually make them interesting and cool. Like, already what's happening is you know, that, you know, it's cool either way basically. You know, we should just keep on with our research, be careful about how we set up experiments, and then, you know, how we do our job in communicating science, because I think, yeah, now it's come to the point that you know, it's hard to convince people otherwise without being too negative about it, which is, yeah, a bit difficult. But I stopped sharing, and yeah, interesting to hearing your, you know, your views on it, and, you know, how helpful you think, as people that I assume do not work on the field, this talk about the Wood Wide Web has been.
Brilliant. Thank you ever so much, Emily. What a fascinating subject. Lots of long words, which I won't attempt to pronounce.
Makes our life
do you see, before we get, I mean, there is a question appeared in the chat box, but I just had one myself, if I may. Back at school, I remember learning all about crop rotation and, you know, the benefits of crop rotation in the context of maximising the output from farmland. Does the science support that or not?
crop rotation. So yeah, that's an interesting one. I am working, even though I'd say like the applied aspects of it is not something that our lab is like strongly focused on. We do try and, you know, do as much as possible, but yeah, we're not agronomists, let's let's put it that way. But I'm working on a British Ecological Society policy document on regenerative agriculture. With crop rotations, the thing is that possibly there are many other benefits. So this is the thing, it's hard again to disentangle. So one can be like a big one that I think has been quite a bit confirmed is the what's called integrative pest management. That when you have crop rotations, you're sort of limiting the amount of you're bringing in more native predators that can eat on the pests that you don't want there. And so
I'm just wondering whether a fungal network, once it's been established and you introduce another crop species, it has to kind of relearn how to synergistically react with the new species.
Yeah. So specifically for AMF, and this is something that it's been looked at time and time again, the specificity of plant species and AMF species. Generally, currently, we don't think they're that specific. They're quite generalist. So one fungal species can actually colonize a majority of plant species. So that shouldn't be a problem. Also, at any time point in your soil, you have many fungal species there already. So you might have, one coming more to surface and being more prominent depending on the plant species that you're cultivating. But yeah, if anything, you're maintaining this native soil biodiversity by doing that, which arguably, yeah, it's a good thing.
Okay, thank you, Emily. Let's move on to the chat box. I don't know if you can see this. First question up here from Malcolm. Emily, I'm interested in soil carbon storage and have indeed been involved with researchers at Reading University. I've read about glomalin as a carbon-rich product of arbuscular MF. Some say it doesn't exist. Others say it's a very important source of long-lived soil carbon. What do you know of it?
Yeah, not too much actually. Yeah, and few of my colleagues are working specifically on the types of nitrogen and the types where it's organic or inorganic sources, and same for carbon, whether it's more in this form of sugars or lipids. Because with the experiments I'm doing, I'm mostly interested in the short term, because based on what I showed you, I only label my plants for 24 hours with radioactive carbon. So I can only account for recently fixed carbon. So I don't know so much about the long term. Glomalin, yeah, I've heard people discussing about this debate, and some people, yeah, presenting it as the go-to answer for anything that we don't know. But I don't know much more, unfortunately.
Okay, Emily. Thank you. Another question from Lee, a general question about fungi. They are composed of eukaryotic cells, but there are a wide variety of cell types in fungi. Are they widely differentiated like other eukaryotic organisms are? Trust you, Lee, to give me a difficult word like that.
Yeah. I'm trying to think. Again, I'm not a cell biologist. We don't usually work on that level. I work more on the ecological sort of bigger picture level. But generally, no, they are not as complicated in terms of their cellular development. So you've got the hyphae, which are in most parts the same. You've got the tips of the hyphae, which do more the exploration. So they tend to divide sort of much more, and they grow much faster, especially when they've got a signal that they're detected. Then you've got your spores, which are again different to that, but yeah, I'm not sure exactly, compared to other organisms. I'd say less, but again, I'm not 100% sure.
Okay. All right, thank you. Stuart, as a follow-up to Malcolm's question, can mycorrhizal networks be enhanced to capture atmospheric carbon?
Yeah.
Or lock it away?
Yeah, I think the general consensus is that they can. I think, again, I don't work on carbon capture. I think from what I know, one common debate is how, again, long term that is, and how much in terms of future disturbance that can actually counteract any effect of locked up carbon. But yeah, there is this, they're called SPUN. It's the Society for the Protection of Underground Networks. It's sort of a recent thing that has been started, and they're trying to map the diversity of fungi that form networks across the globe, sort of, but get a better idea of where we might have hotspots and sort of just generally, understand our below ground biodiversity. But they're also want to tackle this topic as well. But yeah, it will be a big one. Anything that has to do with carbon capture and storage is just too complicated even for the experts. So yeah, I wouldn't want to say too much on that. But yeah, the idea is that these fungi require carbon to live, so they can get a lot of it from the plant, and they do store a lot of it. So yeah, one argument is like a lot of our soil carbon is actually within these fungal networks. But yeah.
Okay, Emily, another couple of points on chat, but I think there's an answer there as well. So I'm going to move to Neil.
Yeah, I'm also interested if other people, I don't know how you usually run these meetings,
Neil.
but obviously I can sort of see from the quality of the questions that you're all very knowledgeable. So if anybody wants to share their expertise and knows better, yeah, I'm happy to.
We go to Neil. He's had his hand up for a long time. Neil, you're muted. If you'd like to unmute and go ahead.
Ah, that's it. Fine. For some reason, it wasn't unmuted. Right. I've actually got 4 quick things, and it might well be best to say all 4 and then do it. I've been using root grow for 10, 12 years, and I'm not sure whether that is a combination of different things or a singular one, and whether there are any positive or things that you should avoid doing, with it was that one. Related to that, I'm one of the things I expected it to do was a general soil improvement thing, and I'm not sure whether that's got any truth about it at all. Then some specific, the other 2 were specific. You didn't talk at all about parasitism, and are there parasites on mycorrhizal fungi, and do any mycorrhizal fungi parasitize each other? And the 4th thing was, have you done things on the length of the hyphae, on things on the experiments you've done? So, did you pick up all those?
Yes, I think I remember. The first one was root grow. Yeah, so root grow. I mean, one thing with these companies, and that's another sort of side debate in terms of application of mycorrhizal fungi in agriculture, because obviously you have companies profiting from this. They're selling stuff. They're being very, even with us, that we use some of their commercial inoculum for scientific purposes. They're very they can be very secretive, about how they do it and what they do it and what's in there, because personally, I don't agree with, but there has been a lot of discussion also about using non-native species, because a lot of these inoculums, they use non-native species. So, what does that mean in terms of preserving our native biodiversity? The difficulty with that is that in many cases we don't know what our native biodiversity really is, because it's just not enough monitoring happening below ground, especially with mycorrhizal fungi. I think there's more and more, like my colleagues who are working on this, what I was saying, newly discovered type of fungus. They're doing a lot of sampling across the Peak District, for example. So we'll have some more results coming in about the biodiversity in the Peak District. But I think across the country we just don't know. So it's an argument. It's like, whether do we don't do anything until we know, or just monitor the situation and see how it goes. I don't know specifically about root grow. They will try and say they're same with mycorrhizal fungi. There's these million benefits. I think it does really depend. Generally, plants do respond, better in terms of agriculture, especially intensive agriculture. The idea is that we can use less fertilizers if we sort of harness more the power of mycorrhizae. But I think you're right. The other important thing is sort of monitoring it a little bit more, the other, in some cases, indirect effects. So with mycorrhizal fungi, one obvious one is the soil structure. So yeah, it's been shown that because of the hyphae, they can increase, well, they can enhance soil structure. So one big problem with soil is that it just becomes eroded, it crumbles, especially in intensive agriculture. You almost get it, it's almost very dusty. Whereas hyphae can have this capacity. And with globally, what we're saying, so been said that it gets released from the hyphae and it almost acts as glue and sort of brings soil a little bit more towards its better soil structure, which is a benefit to all plants really. And then the other big one is the pest tolerance, which I don't think there's been a lot of, and I'm also trying to do that with nematodes and with aphids and with the underground networks. We're trying to see whether there are any specific defense signals being passed below ground by the hyphal network that will enhance the plant's ability to respond to pests. But there's that some people have shown it happens but haven't really specified. They just say signals, so it's still all up in the air. But yeah, these are the three main target areas in terms of crop performance and the other benefits in terms of climate change and carbon storage. Then your next question was,
I can't remember. Neil, are you there?
Sorry,
you're muted again.
I can't see you.
Oh, that's it.
Yeah.
For some reason, it only unmutes on the third attempt. Yeah, I asked about parasites, either on or each being parasitic to each other.
Yeah. So from a plant perspective, with the parasitic aspect, I mean, a lot of people have been trying to view it as a nutritional. So because we focus so much on how much phosphorus and nitrogen is the plant getting from the fungus. If it's not getting enough, then it's a parasite. But is it really? Because there are other potential benefits from a plant perspective as well, as we're saying, in terms of the pest resistance or tolerance. So it depends, what you want to focus on, how you see it. And then there are people that are looking on parasitic fungi, which are they're different category. They're not so much, they don't fall under the mycorrhizal umbrella. But again, I don't specifically work on those. So yeah.
Okay, we must move on. We've got another question from Joe Cork in Bristol. Joe, would you like to unmute? There you go.
Yes, it's hang on. Okay, I have a comment and a question, and thank you for the talk. My comment is that citation bias and over-representation and interpretation of results is probably not something new and has been going on for a long time. It may just be that there's more pressure by the universities to get your stuff out there in the media, which is a pressure on researchers, but it probably isn't new. That's just a comment. My other question is that one of your slides showed your temperate trees and ericas and orchids, all with mycorrhiza, and then you're working on potatoes with mycorrhiza. So do we find mycorrhizal associations in every plant that anybody's ever looked at?
No, it's not every plant. I think, again, depending, how we measure it. I think with all mycorrhizal species, I think it goes up to about 90% of all land plants. With our arbuscular mycorrhizal fungi, it's between 70 and 80%, depending on how people measure it. And we're still discovering that. I mean, and also what is it? It gets very philosophical sometimes, in terms of what is a mycorrhizal fungus. Initially, it was Matthias Rielig, who's the person I was in his group in Berlin. well-known soil ecologist. Yeah, he writes some general blog. He's got a blog as well, so I would point you to his blog entry. So if you click Matthias Rillig Ecologist and you go to his blog, you will see he wrote recently about this. What is a mycorrhizal fungi? And I say that because sometimes, you know, you have endophytic fungi, which can maybe transfer some resources and nutrients, but how much is enough to class them as mycorrhizal? So yeah, it becomes a little bit of a philosophical debate and definition debate. But in terms of vegetables, for example, yeah, because I see a question in the chat as well. The common ones that do not form mycorrhizal associations, we think, are the brassicas. So they do not.
Okay. Thank you for that. Ronnie Jones, your video is a little unstable, but go ahead.
Thank you for the talk, most interesting. Thank you again. Fungi tend to, do fungi and mycorrhizal tend to be selective on the plant or the tree that they would form a symbiotic relationship with? For example, you find certain types of fungi under certain trees, or field mushrooms are only found in fields and not in woodland. Mm-hmm. So presumably field mushroom must form a relationship with grass. Does it form a relationship with dandelions and weeds, or?
Yeah, I think there is some, especially with mushrooms, for example, in terms of where they grow, there is definitely a tree species association. In terms of, again, I'm not a mushroom expert though, so I'm sure there are quite a few people that are keen on mushroom foraging. So yeah, maybe best to ask them. In terms of colonization, some species can be very generalistic. Some are a little bit more specific. It really depends. And we still don't know. We're still matching all the plants with the soil biodiversity, with what bacteria are there, and the environmental conditions, and even the bedrock of the soil, or the soil type. Like all these things are going into the models to see, you know, what is the stronger factor and what is the biggest influence of the composition of biodiversity in the soil. It's very hard to disentangle this. It depends, you know, how you frame your question and I don't think at the time there's any clear. And also what drives what? Like, is it the plant driving the below ground biodiversity, or is it the below ground biodiversity driving the plant composition? Like, this feedback loop is a big debate. A good argument's being put forward. Both.
Okay. As an exercise, I mean, is it possible if you dig up some filaments of the fungus, you're able to tell that filament comes from fungus A and another comes from fungus B?
I think with spores, I think with filaments or hyphae, so much it's more difficult. But with spores, there are some people that can do it. I cannot. I think increasingly, for better or for worse, the younger generation is relying more on sequencing and DNA methods to say what's what, rather than looking at it under the microscope. And there was a big degree, even with the best experts, there was a lot of degree of uncertainty when you look something down the microscope as to being able to say what it really is.
Okay. Thank you, Ronnie. A couple more, and then I think we should start winding up. Robert from Newcastle.
First of all, Emily, I'd like to thank you for an excellent talk. The obvious joke is that it was all Greek to me, but in fact it wasn't. No, as a digression, I'd just say that there's a truly excellent program on mushrooms on Netflix, and I will share the link. Yeah. Right, thank you.
Perfect. Yeah, I think I've heard of that. I haven't watched it yet. Same with David Attenborough's.
It's in the chat box, everybody. Thank you, Robert. Okay, Trevor, that's it.
Oh, it wasn't working at first. Okay, thank you. Thank you very much, Emily. That was very interesting, thought-provoking coverage of the subject. I noticed your interest in understanding what people's knowledge, their awareness and perception of the subject is from the little survey you were conducting. And I would have to say one thing about that, which is that I was interested in following up the recent paper in Nature that you brought to our attention about the controversy, obviously, in the subject. But I noticed that that's behind a paywall, and they want £25 for me to look at it. So I would just say that it's very difficult for people, particularly more lay people, to get to grips with some of these subjects because they don't have access to the definitive sort of research papers and so on. And
yeah. Don't know if you wanted to say. The only thing I can say, yeah, I absolutely agree with you. I'd say one thing, if you're really interested, like drop the authors a quick email. I'm sure they will more than happily send you their articles. Yeah, that's a big debate. I mean, there's what's called an open access now, so they're trying to more and more move towards that. It's a very complicated situation, basically. What's happening is like publish, especially, you know, these authors chose to publish in Nature. They paid a lot. They did the work. They paid a lot of money to get their article published, and then not only that, but then their article is not freely available. And you mentioned that for the general public, which is bad. What I'd say arguably is worse is that there are scientists in other countries that cannot afford these fees, and that's a big problem. I think generally the scientific community agrees. The problem is, what can we do about it? So people who are more influential, they've tried to boycott some publishing bodies, for example, like Elsevier and other sort of for-profit companies that charge large amounts to us to publish, but also, you know, don't make their science accessible. What we try to do is to publish as much as possible to society journals, so journals that belong and managed by the British Ecological Society, for example, or the New Phytologist Foundation. I think that's a sort of easy first step, but yeah, it's a big debate.
Yes, I think the world of academic publishing doesn't need a bit of
I think you raise an interesting point, Trevor, and I'm wondering, in the back of my mind, whether the U3A might itself have a role to play in this, as you know, learning is one of our mainstays, and okay, we don't like to refer to ourselves as a university, but you know, there are many people in the U3A that want to continue academic studies. So some help to access papers that normally, you know, come at a cost, it might be worth looking into.
Yes, that's an interesting thing. I mean, it's something that I already use to some degree. because institutions have subscriptions to many of these journals that can be used by, for instance, their alumni or people who are still associated with them in some sense.
Yeah.
But yes, we might think about the U3A having some role to play. As you say, that's an excellent
Worth a thought.
idea, yes.
Okay.
I think increasingly, hopefully, it is going to become better. I mean, there's more and more universities that are giving more money because basically it comes down to when you publish something now, it's like you pay, say, £6,000 for open access, or you pay £2,000 for not open access. This is the choice that a lot of academics are given. And there's a lot of other things that come into that you need to consider when you publish something. But yeah, no idea.
Okay. Thank you very much for the thoughts there.
Yeah. Okay, I think we have to wind it up now. So I'd like to first of all thank Emily for a marvellous talk this morning. Very thought-provoking. I love the graphics, and yeah, it's a whole new world down there.
Yeah.
So
Thank you so much.
yeah, hang around. Please all thank Emily for her talk. Thank you very much.